Okay, I think we're good to, yeah. Good afternoon, everyone. Today, we're delighted to have Bacem Saada presenting today, who holds a computer engineering degree from Tunisia. He also holds a Doctor of Engineering degree from the Harbin Engineering University in China. Bacem's PhD research focused on genomic data analysis and DNA sequence compression techniques. He is specialized in bioinformatics, data science, and healthcare systems. Bacem worked as a bioinformatician for 2 years at the Laboratory of Microorganisms and Active Biomolecules in Tunisia. Between 2019 and 2021, he was a postdoctoral scientist in the 8th Affiliated Hospital of Sun Yat-sen University in Shenzhen, China, focusing on cancer genomics. Recently, Bacem joined the Animal Bioscience Department as a senior data analyst, leading the activities of the computer group. And with that, Bacem, I'll throw it over to you to begin. Thanks.
Thank you, and good afternoon, everyone. I would like first to thank you for providing me the opportunity to be the speaker today. And my talk will be about genomes alignment, algorithmic aspects. Here is the outline of my presentation. I will start by an introduction, then I will provide some methods of whole genome alignment, and finally we will have a discussion. So let's first talk about the whole genome alignment. The whole genome alignment is done to understand the present. For example, if we have some strength, so is it pathogen or non-pathogen host? It's also about the host specificity, and if there is any difference at genomic level between two or multi-genomes. Orthologous regions, conversion rate, how the genome they are compared to each other. It's also important, the whole genome alignment, to be done to understand the past, how the organism evolved to be what they are now, and to describe characteristic to identify functional element on the actual species related to their ancestors. So whole genome alignment, to explain it, for two genomes, genome A and genome B, we try to find a mapping from each nucleotide in genome A to its corresponding nucleotide in genome B. However, life is tough. Usually the genomic comparison is not that easy. There is a lot of insertions, deletions, translocation, inversions, and duplication. So it's important to know which method we have to use to do the whole genome alignment on a correct way. In the beginning, a few years ago, we were talking about DNA sequences alignment. However, for example, I think that you have knowledge of the Wulsch and Needleman alignment algorithm. It was a whole DNA sequence comparison method. And now when we have the genomes and we have a large size of genomes, it's not, we are no longer able to use these methods. So that's why now we are trying to implement whole genome alignment, which consider the memory problem as the genome size now are very huge and big, and the complexity to know how the complexity of this algorithm in terms of execution time, computational time, and how they are sensitive to parameters change. For the whole genome alignment, there is three classes, three main approaches. The first one is the suffix tree based approach. The second one is an Uncharged based approach. And the third one is a graph based approach. The first class is about suffix tree. A suffix tree represents all the suffixes of a string T. So if we have, for example, for the example that we have, there is more than a suffix tree. It can be considered as here, there is all the element of the suffix tree. Why we use the suffix tree of a string T? It's because we can reduce the search time. For example, if we are trying to look for a chain which started with A, we can focus on this And if it's, for example, we know that the first letter of the chain is A and the fourth one is T, we can, for example, just see this branch of the suffix tree. So if there is any other elements where the first element, for example, the second element and the fifth element are not both A, we will not see on the other parts of the suffix tree. And also it's for the storing size, because when we store the sizes, we do not need to store all the element. We have the suffix tree. So when we have a string, for example, we have the longest one, we will be able to predict all the previous suffix tree that we have for our element. And also it's also for the balance. So if we have an element, we are, for example, in the root, and based on the element that we would like to make the research on the string, we know if we will have to look on the left part or in the right part. So it will reduce the research time for the element that we would like to get to the half to 0.5. The second approach is Uncharged based methods. Usually, we try in this method to detect regions with high similarity or identity. And we try to, if there is some distance or some mismatch between the two regions with high similarity, we can decide if we can merge them and put them together on the same region. The third class is the graph-based homology mapping methods, and they try to make the genome comparison in terms of gene composition to understand the evolutionary and relationship between the different species and the different genomes that we have on our research. The first algorithm that we're gonna talk about it using the suffix tree-based method is MAMR, and the version 1 was implemented in 2004. The MAMR algorithm tries to detect maximal unique match between two genomes. So as you can see here, the algorithm can detect if we have two regions which have the maximal unique match between the two genomes. So we may have other regions, but their score or their match is less than these two elements that we have here. And to detect this similarity, it tries first to decompose the two genomes onto the suffix tree, and then sort all the exact matches that we have on that element, and then sort them and extract the longest possible set of matches which are in the same order. And then when there is gaps between the two genomes, they use Smith and Waterman alignment, which usually was used for a local alignment for small DNA sequences. So for example, here we're gonna explain how MAMR is working. So if we have the two sequences, we try to put them together on the same suffix tree, letter by letter, and then we try to detect the longest similarity that we have between the two sequences. And here it's this chain, which is the exact match between the two sequences. We have only one maximal unique match. When we have, as I said in the step 1, when we have a whole genome, they try to detect all the maximum unique matches between the two sequences. So first of all, they will detect the longest one, and then for the rest of the region that we have, they try to detect smaller unique matches between the two genomes. And as I said, then they will do the Smith and Waterman alignment method for the region which are non-similar. And that's what I was saying. They can use Smith and Waterman alignment to understand if there is some SNP, insert, polymorphic regions, or repeats between the two genomes. It was first applied to tuberculosis strain. So something that I would like to say, if one of you is working on whole genome alignment, Usually, the methods and the techniques, they are verified for some genomes or some species. So it's important to know if we are using one specific whole genome alignment, to know which one we're gonna use and which one we have to use to have accurate results during our research. And then the MAMR, they implemented the version 2.1. In this version, they try to cluster matches together on the same, as I said, in the same cluster. So if there is two matches that they have distance between them lower than a gap length defined by the user, here it's k, so they will be joined together on the same cluster. However, if there is other matches that have distance more than k, so they will just stay away, stay alone. And then they implemented also the version, the MAMR version 3.0, and they added in this version, they added to detect the exact matches which are duplicated on the subject genome. I said in the version 1 that it was about unique matches, but here they start to see and to check what are all the maximal matches, including non-unique region between the two genomes. And in addition, as it's important, they also try to improve the execution time of the MAMR method and the memory usage. And in this version, the version 3, they started to make alignment of human chromosome versus a human chromosome, which was done in about 300 minutes. Recently, in 2018, they added version 4, and here it was more about computational analysis and more about memory, and it was more about memory and storage. So now with the version 4.0, they have a theoretical limit of an input size of genomes of 141 terabyte base. So usually, technically and theoretically, MAMR 4.04 can perform alignment with input sequence of any biological realistic length. So now, as you know, some genomes, they can reach 20 gigabase, 18 gigabase. So with the version 4 of MAMR, any input can be done and any whole genome alignment can be done if we have all the necessary resources to do the whole genome alignment. Second, I will talk about Uncharged based methods. For this, I decided to talk about Ligand. Ligand is a whole genome alignment who try to detect seeds, which are the similar region between two genomes. So in this part is genome 1 and in this part is genome 2, and they try if they can detect some similar region between the two genomes. And then they put them together. They are small seeds which will be then merged to be together. And a chain of seeds, as you can see, is Uncharged. So this is some example of them. And then after having all the region of similarity, they try to map all the region based on the score of mismatching between the two similar region. And finally, they try to do Needleman and Wunsch alignment DNA sequence. It's used for the global DNA sequence alignment. So using Needleman and Wunsch algorithm, they try to make the alignment between the non-matches parts between the two genomes. And also because we don't have only alignment between two genomes, we have also multi-genome analysis. So they implemented the Multi-Ligand Algorithm. And in this algorithm, we have K sequences as inputs and phylogenetic tree, and they try to perform alignment between the two closest sequences to merge them together and then do alignment to the third or the fourth sequence, until finishing all the sequences that we have in our input set. It was tested on a data set of vertebrate species, and the multi-ligand produced most accurate alignment between exon and chromosome between human and chicken on just 75 minutes on a personal computer. Because as you know, we have servers in our department, and it's working fine, and you have the resources that you need to do your research. However, if you have, you can have a huge data. Even with our servers, we cannot do this analysis. But sometimes some other researchers, they don't have access to servers. So one important thing, even you in your future life, or if you would like to do an experiment or alignment on your own laptop or workstation, so it's important to have algorithm which can make analysis on a reasonable time on your personal computer. For example, myself, when I did my postdoctoral position in China, I was working in the Cancer Institute of my hospital, and usually when I was reading papers, the execution time and the application, I mean, it was done on a personal computer or on workstation. I don't know why. Maybe it depends. Maybe because in cancer there is some hospitals or some research institute which work independently than other than other university, or they have where they have big data center. But sometimes it's also important to have an algorithm which can do the analysis on a reasonable time on your personal computer. There is also other algorithm. It's called MOV. So the main use of this algorithm is to identify the conserved genomic regions and to detect rearrangement and the inversions in conserved regions. The step one, it's like the MAMR algorithm. They try to detect the maximal unique matches, and then based on the matches that we have, they try to make a distance matrix to construct a phylogenetic guide tree using a neighborhood joining method. And then when we have more than one multiple unique maximum unique matches, they try to put them together in the collinear region. It depends, as usual, it depends on the mismatch score. It depends on the distance and on the mismatches between the different region and the other part of the genomes. And finally, this genome, if there is some region which are not unique in the entire genome, when we do the analysis, we try to be sure if this region they are unique only on the matches that we have between the two genomes, or if they are not unique on the entire genome. They may be unique on the non-similar region or unique on the similar region. Based on this information, we can decide which outside sequences on its genome will be concatenated into a single region. If they are unique outside the similarity, they can be concatenated and put together as these matches between the two genomes. There is also progressive MOV. It can align the same differentially conserved region, and it consists on calculation of a score, possible configuration of alignment between multiple genomes, and they use homology hidden Markov model to reject random sequence similarity, because not all the similarity that you have in your in the whole genome alignment, they are significant. Sometimes it can be only random sequence similarities. So we need to make some calculation and to see based on, especially on hidden Markov models, and do some statistics to decide if this similarity are due to something or it's only randomly that they are happen and they are here. And also the first use of this algorithm was to a group of 23 finished genomes on the family of into some bacterian family. And when we did this alignment for the 23 finished genomes, the MOVE algorithm helped to identify the core genome and the pan genome of this group. There is also another algorithm which called BLAST-Z, and it was made in 2003. So the main difference of this algorithm compared to the other, that if we have two exact matches between the genomes and the gap is lower than input user parameter k, so that they can be merged together. And the main use of this algorithm is to align the human and mouse genomes. The third class of whole genome alignment is based on graph. I don't know if you studied graph before, but Mercator is one of the best algorithm to do the whole genome alignment. And this genome is able to detect to define the orthologous map construction between in comparison between the different genomes. So how this genome works? This whole genome alignment try to detect clicks between the two genomes. A click in a graph is when we have nodes which are connected. So it's a connected graph. All the nodes have edges together, and they are how to say it? These nodes, and they are connected between them. It's like an electric circuit when it's closed, and they all have connection between them. So if there is click between the different genomes, as here between genome A and genome B, so this region will be considered as similar, and it will be considered as orthologous region between the different genomes. Let's move now to the discussion part. I hope that you were able to understand what I was saying. I know that your background is not more about computer science, but there is an important thing that I would like to say. It's important to know which algorithm you're gonna use. And it's also important to know how this algorithm works. Because based on the architecture that you have, based on what you need as a result, based on how, if you think that there is parameter that you need to make an input or to change different parameter in the whole genome alignment, it is important to see how the algorithm works and to see how the output will be. And if you modify any parameter in this algorithm, how the second output will be different to the first one. And there is also one important, as I said in the presentation, I tried after each algorithm to make one use in which context they used it. So you can know if you use this algorithm for which species of animals, for human, for mice, for bacteria. So it depends on the species that you are using, you can predict if this algorithm you will be able to use it for your genome or not. For example, MAMR4, which was implemented in 2018. So as you can see, he has reasonable execution time. When they did the whole genome alignment of a human between versus chimpanzee. However, for example, old version 3.0, it fails. We cannot make alignment between a human and chimpanzee. Other algorithm of whole genome alignment, they do the alignment in more than 2 days, which is too much. And also, who knows, we need to check the output between the different algorithm, if the input is correct or not. For example, for myself, when I was in China, it was the first thing that we did when we would like to use a tool is we try to download the data set that was done before, and we have to get the same outputs, the same result at the research paper they had. At that moment, we can say and we can predict that if we apply this algorithm with this parameter to our data set, we will have the result that we are expecting to have. But if, for example, we download whole genome alignment and we just try to play with the parameter, or we keep all the default parameters and we get our data set and we do the whole genome alignment, how can we be sure that the result that we have are are correct and we can use them for our next steps of our research? So this table is to compare to give you a little view to understand that there is a lot of methods of whole genome alignment. Some of them they are used for a local alignment between genomes. Some of them they are used for the global genome alignment. Some of them they are used for the homology mapping and to understand the hierarchical whole genome alignment. It's the same to understand the mapping between the genome that we have, to understand how they are in terms of conserved region, in terms of orthologous regions, and to understand if there is similarity only on the genomic level or if there is difference between the species that we are using for our analysis. I was talking a few minutes ago about the use of your personal computer. So here I used, when I was in China, I did a human versus a human whole genome alignment using 4 whole genome alignment tools. And as you can see, there is 2 tools. They have execution time, which is reasonable, less than 4 hours, when, as I said previously, the MAMR version 3.0, it fails for human versus chimpanzee, and it fails also for human versus a human. And the MOV algorithm, it stayed more than 24 hours to do the whole genome alignment. So as a senior analyst, now with the department, sometimes we have emails, our data is too big, or we were not able to do the analysis, or so it depends on the version of the tool, in which year it was done, and which improvement there were. Sometimes there is some algorithm which they make improvement in terms of complexity, in terms of the execution time, in terms of memory usage, but some tools they just try to get the result and the output correct. However, in some servers or in some machines, we are not able to run these jobs and these tools. And why the whole genome alignment is important. I really like this picture because, as you can see here, there is a lot of, it's a beautiful figure. There is a lot of similarity, but they are not in the same region. There is the conserved region. How we can decide if we see that on the gene and protein level, if we see that on the DNA level for the nucleotide. So it depends on the analysis that we would like to do, and it depends also on the tool that we are using. And also in your research, if you do a research while doing, for example, the comparison in DNA level, maybe you will not have the same result as expected or as what they were done as when you do about the genes mapping. So it depends on the analysis that you would like to do. It depends on the result that you are expecting. And it's important. That's why it's really important to see which tool you are using for your experiments. So as a conclusion, I try to present a useful algorithm for the whole genome alignment to detect similarity between regions. And as I said, there were one professor before, and he said that similarity is not the important part of the genomes. We have to understand the non-similarity, the rearrangement, the repeated regions, the copy number variation, the SNPs, and based on the result that we have, we can understand the relationship in terms of evolution between these genomes, especially that now I think that you know that there is a tree of life, and in the tree of life, so we try to make all the species around the world together, starting from the head, and then so they are trying to detect how the evolution make that species were all starting at the same point in the first beginning of our Earth, and then there is that lot of non-similarity and a lot of species that we have nowadays. So the limitation and the problem that we may have. As I said, it's important to understand which parameter we can use to perform the best alignment and to have the best result for our experiment. It's also how should be the length of the similar region between the genomes, which dynamic programming alignment method we can use. So if you remember, we saw that one algorithm used a local DNA sequence alignment for the non-similar region in the genomes, Smith and Wattelman algorithm, and other one was using the global DNA sequence alignment method. So we need to know what's the difference, and it depends on the length of the region. And when we see the output result, we can see how these results are accurate. And as I said, if we compare first we try to compare with existing data sets on internet and saying the output and trying to understand it and saying how the other paper they wrote their conclusion and their discussion. We can, after that, when we do our own alignment, we can see which algorithm would be better for our experiment. We need also to understand which regions to be aligned. If we can, based on the genome that we have, if we can consider two regions with high similarity, we can concatenate them together if there is a distance and mismatch between them which is small, or if we cannot do that. And so that's the same as the other point, how to extend identical regions. With which parameter we can extend the region that they are similar between the two genomes. How to delete and how to detect random genome similarity. Similarity, they can be just random similarity, or it can be real. How to say? It can be real, and there is a cause of this similarity. I talk too much about my journey in China, but sometimes when I was, as I said, I was working on cancer genomics, and it was about immunology. So sometimes when we have population of immune cells, we have some genes that they are present on the whole cells. We can say that it's a random existence. They exist because they are here. And we can say, oh, that's not possible if they exist in all the cells. So it means that they have something to do with the weak immune system related to cancer and tumors. So it's important to understand based on the non-similarity between the genomes and based on the outputs and comparing our work to the work of others to understand how the output, how the result, how this algorithm is performing the analysis and providing us with the output to understand if our work we should continue to go on the same way or if we have to use other method maybe. And also one important thing, the last one, is which scripting language we have to use. In some cases, when we are doing input from files and writing on files, so if we have a large matrix, for example, importing from files and then performing some analysis and saving them later. So Perl usually use memory 8 times less than Python. So if you are having a script and use it in Perl, it may be 8 times faster and using less memory than Python. However, it's not in all cases, but one thing to consider to think about, depending also on your input and the output and the large of data that you have and into the script that is done, which kind of instruction and computation you are having in your script. So it's also important to consider which algorithm to use and which value to use. For example, if you use integer number, if you use number 1, so you define a variable with value 1. It's way different between defining a real or a float value, which can be considered in memory as 1.00000. So there is a lot of memory to store all the data. If we have a lot of data, so we will have a large amount of data on our RAM memory and on the disk. And also if we will do mathematical operation, or if we try to however is our computational what we have, but it's a big difference between we have to decide which type of variable we will define for our script and which programming language we are using to have the less problem in the future about the execution time and the memory usage. This is some references for the presentation, and thank you for your attention. If you have any questions, please let me know. Thank you.
Thank you, Bacem. So yes, if anyone has any questions, feel free to raise your hand or to type your question in the chat and I can read it out for the group. Got one. Flavio.
Okay, can you hear me?
Yes.
Okay. Thanks, Bacem, for the presentation. Well, for me, it was kind of above anything that that I know. But I'm curious about something, Bacem. When you align like two genomes, different species, let's say, right? So the algorithms they do in the individual base, like the genome of one particular individual in one species with another individual, or they use a pooled DNA, right? A genome of several individuals, which is how the algorithm do this.
Could you repeat, please? I wasn't able to hear you well.
Okay, so I wonder if what is the best strategy, right, to align the genomes is to, if you like to align the genome of two species, and so you use one single individual of one species, and then you try to align with a single genome of the single individual of the other species, or you use the genome of several individuals, like in a pooled sample, to try to align. So
Yeah.
I think it's possible to do with these two strategies, but which one is the best one?
Yeah, it depends on how similar the genomes and the species are to each other. For example, a human and chimpanzee, they are similar of about 97% or 98%. So we can, and it depends also on the output, on how if we are looking just to similar region between the different genomes, or if we are trying to detect the evolutionary and the orthologous and paralogs genes between these different species with their ancestors. So it depends on the output, which kind of output you would like to have. But as I said, there is some species that they are quite similar. So we can do it on any way it's possible, like a human and chimpanzee. But for other species, it depends on the output that we would like to have. And if it's only whole genome alignment to see which differences between them, or to understand how genes they moved from one species to another, and how they were similar based on their ancestors.
Okay, thanks, Bacem.
Thanks, Flavio. Do we have any other questions? Okay, I'm not seeing any other questions, so I'd just like to thank you again, Bacem, for the great talk, and we'll see everyone again here next week. Thanks.
Thank you. Bye-bye.
Thank you, Bacem.
Thank you.